Role of nucleus in epigenetics. The nucleus play a central role in epigenetics because it houses the genetic material, that is the DNA, and the molecular machinery responsible for regulating gene expression without altering the DNA sequence. So epigenetic modification in nucleus, DNA methylation. It occurs in the nucleus where the DNA methyl transferase enzyme add methyl groups to cytosine base. So typically repress gene expression by making DNA less accessible to transcription machinery. So histone modification, histone in the nucleus are modified, example acetylation, methylation, phosphorylation to alter the chromatin cell structure. Euchromatin opens and active for transcription, and heterochromatin it condense and transcriptionally silence. It is silent. So chromatin remodeling complexes repositions or restruct nucleosome to regulate access to specific DNA region for transcription. Non-coding RNA, long non-coding RNA and microRNA miRNA are transcribed in the nucleus and influence chromatin structure and gene slicing. Nuclear organization and epigenetics. Chromatin territories. Chromosome occupies specific region in the nucleus. It is the chromosome territory, and then influencing the gene expression. Gene located near the nucleus periphery are often silent due to association with the heterochromatin. So the near nucleolus, it regulates ribosomal RNA synthesis and can influence gene expression indirectly through stress response pathway. Nuclear matrix, it acts as a scaffold organizing chromatin and facilitating or repressing gene expression. Here the role in the development and diseases. In cell differentiation, epigenetics changes within the nucleus guide cell differentiation by activating lineage specific gene and silencing other. So disease mechanism also in that also epigenetics. Dysregulations in the nucleus contributes to cancer, neurodegenerative diseases and other disorder. Role of nuclear pore in epigenetics. If we see the nuclear pore, they are large protein complex embedded in the nuclear envelope, allowing secretive transport of molecule between the nucleus and the cytoplasm. Recent research highlight their roles in epigenetics and gene regulation. Transport of epigenetics machinery. So nuclear pore regulate the import and export of the molecule involving in epigenetic process such as transcription factor, histone modification enzyme, for example, histone acetyl transferase deacetylase, and DNA methylation enzyme, for example, DNMTs. So gene positioning and expression. Gene position near the nuclear pore often exhibit active transcription due to accessibility of transcription factor. Nuclear pore complexes can thread specific gene influencing their epigenetic states. If we see the role in the chromatin organization, nuclear pore interact with the chromatin and are involved in anchoring, activate the chromatin region, maintaining euchromatin state near the nuclear periphery. Stress response. Nuclear pore mediates stress related gene expression by regulating nuclear cytoplasmic signaling pathway that influence epigenetic state. The nucleus is the command center for epigenetic regulation, housing the DNA and coordinating modification that control the gene expression. If we see the nuclear pore, it act as a gatekeeper, not only facilitate molecular transport but also influencing chromatin organization and gene activity through spital positioning and molecular interaction. These dynamic role of the nucleus and nuclear pore are critical for understanding how cells adapt to their environment and maintain homeostasis. Eukaryotic cell protoplasm. Protoplasm of eukaryotic cell show a streaming movement known as cyclosis. They consist of two type of movement, that is the rotational and circular. Rotational movement, cytoplasm move around a vacuole in one direction, for example in hydrilla, or that is the leaf cell. And if we see the circulation movement, the movement in different direction around the different vacuoles, for example staminal hair of Tradescantia, and these movements are due to the colloidal nature of the cytoplasm and microfilament activity. If we see the nucleus, it is 1/10 of the volume of a cell occupied by the nucleus. In a cell, there is a different nucleocytoplasmic ratio. Nucleocytoplasmic index is volume of nucleus upon volume of cell minus volume of nucleus. About 10% of the nuclear membrane bears simple or compound pores. Cell may be unicellular or multicellular. If a multicellular condition arises due to fusion of cell, it is known as syncytium. Example, plasmodium, body of slime molds, young xylem vessel, and if due to repeated nuclear division without cytokinesis, it is known as coenocytic. Coenocytic, that is a eukarya, rhizopus, nucleus. These are some of the example of it. A nucleosome is an octamer of histone protein and has a core of 8 molecules of histone protein, in each of which consists of H2A, H2B, H3 and H4 bounded by 13 by 4 turns of DNA having about 166 base pairs. H1 histone does not form nucleosome. The size of the chromosome varies from 0.5 to 32 micron. The minimum number of chromosome, that is n is equal to 2. Example, Heplopappus, maximum number is 2n is equal to 1262 in adher's fawn, and in animal minimum number is 2n is equal to 2 in Ascaris species, and maximum number of 2n is equal to 1600 in Radiolarians and Oleucantha. Nucleus has 80% of the protein, that is 62% of the non-histone molecular weight high, and rich in tyrosine and tryptophan acid, and form enzyme, and in 15% of protein are basic and histone protein molecular weight low and rich in lysine and arginine. DNA is to histone ratio is 1:1, and nucleosome as structural unit of the chromatin term was given by Outdate. Cell organelles. Cell organelles organelloids are of 4 types on the basis of the membrane. Organelle bounded by a single unit membrane called microbodies, that is a peroxisome, glycoxysome, lysosome, and Golgi body, etc. If we see the organelle bounded by double membrane, they are the plastids, mitochondria, and nucleus. Organelle bounded by the triple membrane, that are the transosomes. Organelle without any membrane, they are the ribosome, centriole, and nucleus. These were some of the important point about the cell structures which was left in the previous video. So I covered here. Thanks for watching. Do like, share and subscribe, and follow by Mindscape for more video.